Thank you very much for this invitation to present our work. We have known for decades that structural variants are important in cancer, starting with Janet Rowley's pioneering work showing that the Philadelphia chromosome is actually a reciprocal chromosomal translocation, and we know that translocations can activate oncogenes. Oncogenes can also be activated through amplification, and were directly observed double minute chromosomes. Double because they show up as pairs, minute because they're tiny in relation to the chromosome, but they're actually circular DNA molecules, millions of base pairs in length. Another mechanism to amplify oncogenes comes from work of Barbara McClintock and Mays, where if a chromosome loses its telomere, then when it replicates, those chromosomes fuse. Now you have a chromosome back with two telomeres, but has two centromeres. And then it pulls apart during cell replication. If there's an oncogene there, cells that acquire more copies of the oncogene will grow, leading to oncogene amplification. So for identify structural variants, we need the highest quality cancer genomes. So for about 2 years, Isabel Rodriguez in my lab has been optimizing methods. We settled on using standard QIAGEN extraction, selecting with blue pippin to get rid of molecules below 15 kb, and generating genomes from a single flow cell with 30x coverage and 50 of over 30. More recently, we've gone to ultra-high molate extraction using a protocol from the MEGA lab. And here we can, this is a simpler workflow, and we can get the same coverage with longer reads. All of this is to feed into the software. We're extremely fortunate to be working with Misha Kolmogorov's group, who has developed a suite of tools that's the best identifier of structural variants that are somatic. So here we generate phased genomes, feed those into phased alignments, and then call structural variants, even very, very complex variants involving multiple fragments. So we've applied this in one case to cervical cancer, looking at the most frequently amplified oncogene, YAP1, and showing that it's amplified using this breakage-fusion-bridge cycle. And I believe we're the first ones to use long reads to sequence across here and identify the structure of what's exactly happening at the position of these breakage. We've also shown that these amplifications define a subtype of cervical cancer that's more aggressive. Patients get diagnosed about 12 years earlier. This is more common in African American and Asian women than in women of European origin. And Emma Robinson in the lab has shown that these women have worse prognosis. So this is a subtype of cervical cancer that's more aggressive with poor outcome, more common in minority populations. We've sequenced a panel of cervical cancer cell lines, have 6 of those cell lines that have YAP1 amplification through this BFB mechanism, and have characterized the detail of those structural rearrangements. We've also shown in the TCGA collection of cervical tumors that virtually all the tumors with YAP1 amplification have this characteristic profile of BFB with a focal amplification in the YAP1 region and then lower copy number in the sequences beyond there out to the telomere. We were surprised then when Natalie Campos, a summer student in the lab, identified a identified amplification of YAP1 in an HPV positive head and neck cancer cell line that was not BFB, but actually involved a circular molecule predicted to be of 500 kb. We can sequence, she amplified and sequenced that breakpoint by Sanger sequencing, but we don't know is it a circle of extra chromosomal circular DNA, or is it a concatemer of molecules? And actually, Sergey has told me just yesterday that we can use POR-C to figure this out. We went back and looked at the TCGA tumors and found 5 of them where the copy number before and after the amplification were identical, which is not consistent with BFB. One of those is due to a breakage-fusion-bridge, but the other 4 are not. So it's about 90% of YAP1 amplification is through that mechanism, and then other mechanisms result in amplification. One of those is a tumor which has the same predicted circular structure with a 2 megabase fragment of chromosome 11. Now, there's a wide spectrum of structural variants that you can identify. Insertions, deletions, duplications, obviously, but then many more complex structures which occur in cancer cells, which the Severus package will identify. But we don't know a lot about when these events happen and do they happen together. So we took 6 cases where there was breakage-fusion-bridge events with amplification and looked for structural variants in that region. And we can find clustering with nearly every complex breakage type except tandem duplication showing clustering together with BFB events. So a lot of these structural variants are occurring related to each other in the same region of the chromosome, and one of our future goals is to understand this more deeply. So here's a case where there's BFB events shown in the middle there, and there are multiple inversions, deletions occurring in the same region. But obviously going to be a big challenge to figure out, do these all happen during the same crisis, or do these happen sequentially over time? Daniela Kayembe, another summer student in the lab, characterized a very interesting tumor with HPV integration right near YAP1 amplification, but then multiple rearrangements between chromosome 11 and chromosome 15. We could identify the initial HPV integration in chromosome 11, but later rearrangements to chromosome 15. So here at least we can identify some sequence of event where HPV integrated into chromosome 11, and later on with these rearrangements with chromosome 15. Finally, we want to go from the structural variant, obviously, to the function. So it's great to identify the structural variant, but we want to know what is the impact on that, on the epigenome, on the chromatin structure, and also on gene expression. And so we're using the direct RNA sequencing protocol on our cell lines to generate optimum transcriptomes to then be able to fully characterize the tumor, hopefully leading to the identification of new options for therapy. As an example, the YAP1 oncogene, which we're very interested in, has at least 12 different isoforms, making 12 different forms of the protein. And some work has been done on the function of these different isoforms, but through the direct RNA approach, where you take RNA, you make a strand of cDNA, but then you ligate an adapter onto the RNA strand, and it's the RNA that goes through the pore, we're able to get very high output and very accurate aligning to be able to identify all these different isoforms, count them up, and then start looking what happens between immortalized cell lines, cancer cell lines, YAP1 oncogene cell lines. The initial result is it looks like if the expression goes up, all the isoforms go up together, which is interesting. But obviously now we can look at this across all the genes expressed in a given cell line. Of course, methylation data, we've heard a lot about deriving methylation data from a nanopore sequence. Here's an example with an imprinted gene where you can see one haplotype is methylated and the other is not. And we also want to look at open chromatin to see what chromatin is active. Winston Timp's group published a paper where they used a methylase to methylate guanine. We're using a protocol that Cecil Jewell and Phil James have identified here with E. coli EcoG2 methylase that generates 6-methyladenine. And here we can see open chromatin regions in the promoter region of a gene that's highly expressed. So in conclusion, a complete understanding of structural variants is critical to cancer diagnosis and therapy. New tools allow an unprecedented view of structural variants and their implications for gene regulation and dysregulation. And I'd like to thank the amazing team behind this that did all the work and all of our collaborators and you all for your attention.